Okay, so in this next video, we're going to pick up where we left off with this transverse T-tubule system with the axial tubules, and now we're moving on to the role of the sarcoplasmic reticulum within the cardiac muscle cell. The role is the same as in skeletal muscle. It's a warehouse for calcium ions. The amount of force we can generate is related to the amount of calcium concentration in the cytosol, just like before, for both skeletal muscle and cardiac muscle. So the more calcium we have into the cell, force will be force will be increased greatly. We just have a better way of transmitting signals in cardiac muscle cells, so we can change the concentration of calcium much more quickly. Okay, so let's talk a little bit about calcium. Calcium is critical to contractility. Two ways we can use calcium to initiate contraction. One, we use the calcium from our sarcoplasmic reticulum. Two, we use the calcium from the extracellular space. If we remove the calcium concentration from the extracellular fluid, we will have a dramatic decrease in contractility and may possibly induce cardiac arrest. An increase in the extracellular calcium enhances contractility, but if we increase it too much, we could also induce cardiac arrest. So we got to hit it in that sweet spot where we have to increase it, but not to an extreme point. Calcium enters the cell in phase 2 of the transmembrane action potential. This is our plateau phase. So go back to your last notes in the last lecture when we look at the cardiac action potential, steps 0 through 4. During phase 2, that's when we're kind of at that plateau phase. That's when calcium enters the cell. All right. So calcium comes into the cell from 3 areas. One enters at phase 2 of the transmembrane action potential because there's an increase in the permeability of the sarcolemma to calcium during that phase. Because there is more calcium exteriorly, it will move down its concentration gradient and enter the myocardial cell into the cytosol. 2, calcium will enter through the transverse T-tubule system, so they'll come down those transverse T-tubules as well. And also 3, from something called a glycocalyx protein. This is found along the sarcolemma, along the top of the sarcolemma, and it also has calcium bound to it. So that glycocalyx protein has calcium bound to it, and it releases that amount of calcium into the cell too. So calcium in cardiac muscle cells comes from 3 different areas. Now we're talking just about the cardiac muscle cell here. Okay, so now where we know calcium comes into the cell from 3 areas, we're going to talk about a concept within the heart called calcium-induced calcium released. Right? So during phase 2, as calcium enters the cell through the sarcolemma, through our transverse T-tubules, and it's attached to this glycocalyx protein, will release its calcium, has bound to it. All 3 of those calciums where it's released is coming from the interstitial fluid, right? From right outside the cell. It is the most important with regards to initiating contraction, but it is not enough to actually activate contraction. So the increased calcium concentration that's now within the cell, so we had just dropped it off from 3 areas, right? 1, moving down its concentration gradient, into the transverse T-tubule system, and 3, the glycocalyx protein that is found along the sarcolemma, which has calcium attached to it, will release its calcium. So we have 3 different types of concentrations of calcium coming into the cell. So with that combination of all those 3 coming in from the extracellular space is enough to initiate contraction, but not to actually activate contraction, right? The increase in calcium concentration that's now within the cell from those 3 locations is what triggers our SR, right here, our sarcoplasmic reticulum, to release its calcium. So calcium is going to be coming in, moving down its concentration gradient into the cell, right? Calcium is also going to be coming down these transverse T-tubule system, and then we have this glycocalyx protein that's along the top that has calcium attached to it. It will release that. So all three of those will release its calcium or move into the cell and will initiate contraction. That increase in concentration of calcium from those three areas now signals and activates our calcium storage from the sarcoplasmic reticulum to release its calcium into the cell. So now with the combination of 1 through 3, as well as the calcium released from the sarcoplasmic reticulum, the concentration is now high enough within the cell to induce a change in contractility of the cardiac muscle cell. This is what is called calcium-induced calcium released. So our calcium-induced section are those three areas of calcium where it's coming into the cell, which causes the activation of the sarcoplasmic reticulum to then calcium release the sarcoplasmic calcium stores. So that's why this is called calcium-induced calcium released. Okay, so anything that causes calcium concentrations in the cytosol to increase enhances the development of tension. Anything that causes a decrease in calcium concentrations in the cytosol, there will be a reduction in the amount of tension generated by the myocardial cells. So for example, our catecholamines, these increase the movement of calcium into the cell. And these are epinephrine and norepinephrine. Another example would be anything that increases extracellular calcium concentration will cause an increase in intracellular calcium concentration because the gradient will be wider. If the gradient is wider, then more calcium ions will enter down the gradient. Anything that decreases the extracellular calcium concentration, we will decrease the intracellular concentration because why? We are decreasing that gradient. Okay, so let's talk a little bit about calcium during diastole, which is the relaxation period during the cardiac cycle, right? We have systole and diastole. And diastole is our filling or our rest period of the cardiac cycle. So during diastole, which is also called ventricular relaxation, calcium has to go back where it came from in order to be ready for the next systole. Makes sense, just like we talked about with potassium and sodium, potassium, all have to go back where they came from. Calcium has to do the same thing. So to get calcium out of the cytosol, we will do that in two ways. One, the membrane itself, so the cardiac myocyte, will use a pump system utilizing a sarcoplasmic reticular protein called phospholambdin. This protein will facilitate gathering the calcium into the sarcoplasmic reticulum. Two, we will move calcium from the intracellular space to the extracellular space in two other ways. One, our sodium-calcium exchange system. So we move 2 sodiums into the cell for every 2 calciums out of the cell. This is considered a electroneutral exchange because you can see here calcium's got 2 positive protons and 2 sodiums are also 2 positive proteins. So that's an electroneutral exchange. And B, our second one is our electrogenic pump. This one moves 3 sodiums in for every 1 calcium out. So we have a couple different pump and exchange systems where we can pump calcium back out of the cell very quickly. But our big one is up here, is this phospholambdin one. So this is another pump system, this is a sarcoplasmic reticular protein which facilitates gathering of calcium back into the sarcoplasmic reticulum. So this is a difference on how quickly this goes and how important it is in cardiac myocytes. Phospholambdin is not found. This protein pump, we didn't talk about this with skeletal muscle tissue because phospholambdin is a protein that is only within cardiac myocytes. You will not find this sarcoplasmic protein pump within skeletal muscle. This is special to cardiac myocytes. Okay, so now how we talked about what enhances contractility, we're going to talk a little bit about our mechanical model. This diagram should look very familiar because this is the same diagram we went over when we talked about skeletal muscle tissue and the contractile elements that are with it. It's the same thing with cardiac muscle cells. So we're going to go over quickly the components again, but hopefully this should just be a review. right? So our contractile components, or our elements, or contractile elements, this is referencing our relationship between actin and myosin. So all contractile elements are attached to a series elastic component. So when we contract our contractile elements, we stretch the series elastic component. So our series elastic element includes tendons and cross bridges of muscle fibers. This is the most component for translating stretch to force. And we have our parallel elastic component over here, right? So this includes our collagenous structures such as our fascicular membranes. This provides stability and protection for the muscle cell. As stated earlier, we cannot overstretch our cardiac muscle cells because that will be detrimental. Our parallel elastic components prevent this from happening, this one right here. Only way we can stretch our cardiac muscle fibers is to put more blood into the heart, right? As we put more blood into the heart, our parallel elastic components become progressively more resistant to stretch as it stretches towards that 2.6 micrometers. So this right here, this parallel elastic component or element, is our protective device for overstretch, right? So our operational window for cardiac myocytes to be stretched is that 2.2 to 2.6 micrometers. As we stretch that muscle fiber, so as we fill the heart with more blood, we're going to put that increase in fluid is going to put a stretch on those muscle fibers. So the more fluid in, the more stretch we're going to put on those cardiac myocytes. However, cardiac myocytes have an upper operational limit, which is that 2.2 to 2.6 micrometers. So as we progressively put more blood into the heart, we progressively become more resistant to stretch. And that's our protective mechanism. And the PEC is what does that.